So moving on to our next speaker, it's a real pleasure to introduce Professor Tony Maxwell, who's a group lead at the John Innes Centre. The Maxwell Lab investigates the structure and mechanism of DNA topoisomerases and associated proteins. Tony's working to further our understanding of key biological processes in which they're involved and to harness this knowledge for the development of therapeutic agents, specifically antibiotics. This work's carried out within the Molecules for Nature Institute strategic program, and Tony's also involved in the Biochemical Society Seminar Series. Thank you for joining us, Tony. Over to you.
Thanks, Jonathan, and thanks particularly to Martha for keeping the ship afloat under these difficult circumstances. So, as Jonathan has said, I'm going to focus on using DNA gyrase and bacterial DNA topoisomerase 4 as targets for novel antibiotics. Now, most people will be familiar with the fluoroquinolone antibiotics, which have been talked about in a few presentations today. For example, ciprofloxacin, that's widely used in urinary tract infections and traveler's diarrhea, and moxifloxacin used in TB. And these compounds have been used for around 40 years now, and they target the type 2 topoisomerases from bacteria, namely DNA gyrase and DNA topoisomerase 4. And although they have homologs in eukaryotes, human topo 2, for example, they're sufficiently distinct that the eukaryotic members are targeted by anti-cancer drugs, whereas we can target the prokaryotic members using antibiotics. What these enzymes do is that they control key biological processes such as DNA replication, shown on the right here, or transcription, shown on the left. Now, these processes lead to topological problems in DNA, like supercoiling ahead of replication fork, and catenanes behind replication forks, and supercoiling in front of and behind transcription complex. And topoisomerases have evolved to resolve these issues. So these reactions can be summarized by simple cartoons, whereby the enzymes can convert relaxed and supercoiled DNA, knotted and unknotted DNA, and catenated and decatenated DNA. Now, all these reactions can be done if you can somehow pass one double-strand segment of DNA through another. And that's exactly how these enzymes work. This cartoon of a type 2 topoisomerase shows a dimeric enzyme binding two segments of DNA, one that's going to be broken and another one which is captured and passed through. And that mechanistic feature allows you to carry out all the reactions of topoisomerases that I showed on the previous slide. And this is illustrated by a cartoon model here showing the enzyme breaking the DNA in both strands and passing another segment through that break. Of course, this double-strand breakage mechanism is extremely useful, but also very dangerous. And anything that can trap the DNA in this double-strand break form is a potential antibiotic. And this is exactly how quinolones work. The details of their action, though, was only resolved from X-ray crystallography by two groups, Mark Fisher, Mark Sanderson's group, and also GSK. And what you're seeing here is X-ray crystal structure of a complex between a type 2 topoisomerase. It's actually a topo 4. It doesn't matter which enzyme it is, particularly it's from Streptococcus pneumoniae. But because these enzymes are so large, what workers tend to do is use a subsection of the enzyme. This is the, if you like, the C-terminal domain of the gyrB part and the N-terminal domain of the gyrA part fused together and bound to a piece of DNA shown in green. And you can probably just see here in space filling mode that these are molecules of moxifloxacin, which are embedded in the enzyme and bound also to the DNA. That's rather clearer from the structure from GSK. This is a different enzyme. It's Acinetobacter topo 4, and you can see the DNA bound across the active site here with two molecules of moxifloxacin embedded into the enzyme DNA complex. So we know in molecular detail now how quinolones work. However, quinolone resistance has become a significant and increasing problem. And this slide shows a correlation between the consumption of quinolones in 3 countries, Greece, France, and Sweden, and how that correlates to resistance to fluoroquinolones in E. coli isolates. And broadly, the more quinolone you use, the more resistance you get. And for these data, it's around about 30% for Greece, for example, but there are now countries in the world where resistance is up to 100%. So obviously, we have a significant problem with quinolone resistance. If that wasn't bad enough, we also have the problem of quinolone-induced resistance to other antibiotics. And this is work we've done recently where in a rather simple way, you can expose bacteria to sub-MIC doses of quinolone, in this case ciprofloxacin, and plate it onto other antibiotics. And what you find is that with quinolones and only quinolones, we find that we get acquisition of resistance to non-quinolone drugs. This includes ampicillin, chloramphenicol, kanamycin, and others. And this diagram basically shows that you can try that with any other antibiotic and you only get resistance to self, whereas quinolones, you get a range, resistant to a range of other antibiotics. So there's an additional problem with quinolones. And so we need to look for other compounds. Now, one way of doing this is through large consortia. And recently, I've worked with the ENABLE consortium, an EU consortium with 28 partners, including big pharma, SMEs, and academics. And one of the programs that we carried out was with GSK, and GSK came with some new compounds and also, very importantly, a crystallography platform using Staph aureus gyrase. In this case, it's the C-terminal domain of gyrase B from Staph aureus fused to the N-terminal domain of gyrase A, and those parts are shown in the different colors here. And as before, we have DNA embedded in the active site. And this forms a platform for crystallography with various compounds. The compounds they brought to ENABLE were the thiophene, so-called thiophene compounds, and 2 examples are shown here. But in summary, what these compounds do is essentially the same thing of quinolones. That is, they inhibit supercoiling by gyrase, but they also stabilize this cleavage complex, which is the mode of action of quinolones that lead to cell death. But most excitingly, what we found with GSK was that these compounds don't bind in the quinolone binding site, but bind in a completely novel site, which is between the gyrA and gyrB subunit in a so-called hinge region. So in summary, what the thiophene compounds showed was very good potency in vivo and in vitro, this very novel binding site, and absolutely no cross-resistance with quinolones or other gyrase-specific agents. And their mechanism is slightly different. We get both double and single-stranded breaks. However, the program was stopped because of mouse toxicity. So to progress this, we've been working with Colin Fishwick's lab in Leeds, where they've been doing medchem, starting with the thiophene compounds GSK1 and GSK2 I just talked about, using Sprout software to do the computational medicinal chemistry, then chemical synthesis, and then we test the activity against gyrase and topo4, work out the mechanism of action and the structure of the complexes, and then feed back that information to the medchem team. So it's become an iterative exercise. So briefly, hundreds of compounds, or at least over 100 compounds have been made in the first few months, and we've been assessing them. And if you look at the structures, we've taken initially the left-hand side of the GSK compounds and made a number of different modifications as governed by the computational med chem. And I've just picked out a few features. So, for example, compound 113 starting to show some reasonable efficacy against the bacteria. Compound 114 shows an in vitro activity that is comparable to the GSK compound, and compound 360 is now also showing activity against topo4, the sister enzyme. So we have the possibility of compounds that are dual targeted to both gyrase topo4. So that program has only recently started, the last 6 months, and we hope to generate compounds which have comparable efficacy but have reduced toxicity. So what I've told you is, although quinolones are highly successful, resistance is a big problem. They target both gyrase and topo4, so that's a real advantage. You get potentially a double targeting. The ENABLE consortium identified these new thiophene compounds, which we've now taken over and using medchem, biochemistry, biophysics, and crystallography to give improved activity. So I'll just finish by acknowledging the people in my lab involved, our collaborators, and our funders. Thank you.